Okay, welcome. Please take a seat. I hope you enjoyed the break, you enjoyed a good keynote, and I hope you are motivated for the next three talks happening here in this stage. Because next on stage, we will hear a talk about AI-driven biochemistry, and I welcome on stage Ariane Mora from AITHYRA. She is a computational biologist and a machine learning researcher, and she specializes in the development of AI-driven methods for enzyme engineering. And with that being said, I am very curious what we will learn in this talk. I hand the stage to you. Thank you so much for being here, and the stage is yours.
Awesome. Okay. I honestly think I can just shout. How does that sound for everyone? Yeah, nice and good? Okay. For those who can't be here. Okay, yeah, thank you so much for the introduction. I'm a new PI at AITHYRA, which is a research institute that just opened up in Vienna, and we do machine learning and biology. So today I'll talk to you a bit about machine learning and biology on one of my favorite topics, which are enzymes. So enzymes are really cool because they're the proteins in our body that do chemistry, or the proteins in nature that do chemistry. And what my research focuses on specifically is how can we find these proteins, how can we find these enzymes that do chemistry that they're not naturally able to do? So how can we find some promiscuous activity and then harness it for things that we might want to do that maybe nature hasn't evolved to do itself? So for example, we could find environmental remediators. So if we're putting toxic pollutants in the environment, I'm just going to set my time. If we're putting toxic pollutants in the environment, we might want to break them down as well. Ideally, we wouldn't put them in there in the first place, but we can't solve everything. We might want to accelerate drug translation, so a way that we can find out how our body's going to interact with an unknown molecule. Design new antibiotics and develop new materials. So all these reasons are why we'd want to design these things called proteins. And so as we heard about in the keynote lecture today, one of the most important things that you have to look about and think about when you're designing a machine learning model in any field, in particular in biology, is the data sets and the evaluations that you're going to do. So the first thing I'll talk about is an evaluation that I did with Jason. This was work I did in my postdoc actually while I was in the Arnold lab. And to introduce you to the data that we're working with enzymes, we want to look at a reaction, so some sort of chemical reaction, a transformation going from substrates to products. And then we also want to have an enzyme. So here I've shown you the 3D structure, which many of you might be familiar with. What that is represented by is a 20 amino acid language, so a different language that they use to the language of humans. And again, this chemical space also uses a different language. So we can encode those in, again, letters, but they're different letters to the ones that the proteins use. Now, when we consider the data that's available for use, that's publicly available, we don't have a nice vocabulary like we do in English. There's the language of the proteins, which we can sequence, so we have a good vocabulary for that, but the one for chemistry is a bit harder, so linking these becomes challenging. One of the other challenges is that we only sample things that we know about, so we only really sample and maintain those positives, and this is a big problem in science. So we don't have as much information about what reaction an enzyme may not do. And so the other challenge is that a lot of this data is collected ad hoc and can be low quality. So for example, if we were only scraping data from Reddit boards, that might be not making the best machine learning model than if we also took all the books that were the high quality literature of the world. So one of the other challenges is also how do we want to actually have an output of our program? So what is the evaluation? And in existing models, so this is already almost outdated, but what they did was they did a hierarchical encoding classification scheme, which takes a broad scale class down to a specific chemical reaction that we might be interested in. But this chemical reaction might not be as nuanced as if we were actually taking the language of chemistry. So that's what the newest models are trying to do. But it's a much harder problem because we don't have a complete data set to train on. So, and assuming most people in this room aren't biologists, so I also came from a computational background, and I always like to show a bit about how we actually collect the data. So one of the things that my group focused on, and this is work that I did in my previous lab as well during my postdoc, is really how do we collect data more efficiently? So if we've got this problem that we don't have enough data, then we need to collect more, right? But then in academia, we have this problem of money. And so one of the better ways of collecting more things is to reduce the cost. So for example, in a directed evolution campaign, which is a way to engineer enzymes to have new activity, you'll collect data, you'll make a data set. Now this data set is created through a stochastic process, And before we actually run our experiment, we don't know a priori what the sequences in here are. So we've essentially applied a random process to some E. coli bacteria and created a new set of sequences. And so the existing methods, basically, you'd only sequence or you'd only capture the data of about 4 to or a couple of % of your data set. And that was to do with costs, essentially. And so we made a way that we could reduce the cost significantly by using a new device. It's a handheld sequencing device. So that basically you could collect all this sequencing data for about the same cost as initially capturing just the top few variants. So we thought that was pretty cool. So you could collect both the positive and the negative data for the same price, which means we're now starting to store and capture that information. When we think about capturing and storing, we also built a database on this. I won't talk much about this, but one thing I just wanted to highlight was we kind of took this shift. The gold standard in the field typically used to be the manual curation of data, but if anyone's done manual curation, it's kind of horrible. And we usually work with master's students and they have a terrible time doing it. So actually we had a master's student who came actually from Vienna. He was in the Defense Force, Pierre. And he was like, how about we try and do agents? So with Yuming, we decided to try and use agents to collect this data rather than humans manually doing it. And then we did the manual check on the agentic extraction from the literature. The models weren't as good back then, so we only actually got 30% accuracy, but it still managed to cut down the time from like 5 hours per paper to just 20 to 30 minutes, make it significantly more efficient to capture high-quality data sets using a combination of manual and agentic-based extraction. So I know I was building up a lot about the data and whatnot, so I'll talk a little bit about one of the models that we built using this data. So in biology, again, we might have this complex molecule, so the protein or the enzyme as a whole, but what we really care about is not necessarily what's going on around it, what's happening at the catalytic site. So when we're thinking about the chemistry that's occurring, most of what we care about is occurring in this small region of the protein. So you can imagine we want to extract the signal from a significant amount of noise to know what are the effects, or I guess, what is the most important contributing factor when we're trying to design new ones. So together, this was work done by Will, a master's student at the time, who's now a PhD student at the University of Queensland. And we developed a model that aimed to essentially take an amino acid sequence, transform the per token embeddings. So for a sequence, you get embeddings per essentially letter. We wanted to learn a new representation that was able to capture the nuanced information of the catalytic residues, not just of the protein sequence as a whole. Feed that through some sort of classifier, LSTM, could be anything, and then so that what we get on the output is essentially a sequence, but now with an annotation to each of the residues as to whether they're catalytic or they're really important for our particular project. So we wanted to know this importance as well because it enables us to engineer them much better. So to do this, we developed this architecture, and I always think it's really, I think also one of the things that came out in the talk earlier is it's not necessarily the best architecture, but how we define our problem and how we define our data. And for us, in our ablation studies, what we found was most important in the architecture was the design of this contrastive loss pair scheme. So for biology, when you're trying to maximize these hard negatives, you have to think about, I guess, the factor that's most abundant in your data set and then the thing that you want to maximize the most. So for us, we needed to pair our amino acid residues together and then try and separate them based on whether they were catalytic or not catalytic. And so I think this was a cool way where we could use our biological understanding of the problem to maximize the information content that the machine learning model could learn. And we used an ensemble of these estimators of these pairs, essentially, because we're never going to, I guess, capture the entire space when we're just doing a sample. So by taking multiple random samples, we were able to improve it as well by capturing that in just taking the variance of the models. And so we need good evaluations, and the evaluation that we used was this benchmark. This is a standard benchmark in the field that was existing from another paper. And so I'm just putting down the existing sequence-based tools, which are a little old now. And then we have some tools which really improved performance because they started capturing that structure. So what I've been talking about is the sequence, so the amino acid sequence, but you could think about capturing that 3D representation as well. And so these are two structural-based methods. But this next one is like a very simple sequence-based method from actually the 1990s, so quite a while ago before machine learning. And it's incredibly effective still. So one of the things in biology is we're at this stage where we still need to use those simple-based models as a comparison because they perform very highly. And what we realized was our model performed on par as well. That got us thinking, and it was here we were asking the question, if all the models are performing the same and we're kind of saturating at this point, is it because the benchmark isn't really testing what we want it to test? So in biology, what we don't really care about is if you've got a sequence or a sentence that's exactly the same, I kind of already know its function. We know what those words are going to say. What we care about is understanding a completely new sentence. So we needed to separate this data set out to make a benchmark that captured how our model could perform in understanding a sentence when it wasn't similar to the training data set. Because what we realized was this existing benchmark that had been previously published had highly similar, I guess, test sets to the training set. So we made this new benchmark, and we could see that the performance significantly dropped. It also significantly dropped as expected for the similarity-based one, but nicely, we could see that now our structural-based machine learning classifiers performed higher, and yeah, about on par with our sequence-based one. So those were two of the contributions from that paper. Now I talked a bit about, I guess, activity of sequences, how we'd want to essentially find this new chemistry, and a little bit about finding particular positions within a sequence that have important pieces of information in them. Beyond that, what we really want to do is move towards understanding, which none of these models have done, but things I hope to do in my faculty career. And so that we can move beyond to ask, I guess, broader questions. Can we move beyond simple classifications to say, hey, this number is associated with this sequence, to really move to ask, okay, how are these electrons moving? How are these products being formed from this complex substrate reaction set? And importantly for us as well, because we're considering about enzymes which don't function in isolation, how is the context or how is the environment that this enzyme is in impacting its activity? So for example, if we're putting an enzyme in laundry detergent, how is it functioning when it's in your washing machine? Or if we're using an enzyme to break down pollutants, how does it function when it's in the natural environment? And so I'll just take that to a bit of the stuff that my group is doing. Some things that we're looking into is how do we then take these sorts of information to expand our knowledge about what biology can do? How can we use that understanding to understand the mechanisms and then move beyond what biology can do to new things in chemistry? And we're trying to look at this in the context of things for human health. With that, I always like to appreciate the open source tools because in computational biology and in bioinformatics, you know, it's not just one tool, but typically you need to use a whole suite. So we've kind of been collecting these and we're trying to consolidate some of these tools and really appreciate the effort that people put into the open source community. The Hugging Face stuff was really cool because of that too. And with that, I really like to thank my postdoctoral lab in California and then also my new lab members who have recently joined the team, Gabriella, Sebastian, Luca, Manuel, Will, whose work you saw before, and Akumi. And with that, any questions, no matter how small about biology, I'm always happy to answer. Cool, thank you.
Thank you so much. And with that being said, are there questions? Who wants to know something?
It can also not be about biology, that's cool too.
Oh yeah, I see a question in the last row.
Thank you. So my question would be, how was your class balance between like positives and negatives? Because I can assume that there will be lots more negatives than positives. And how did you like try to tune it to make it work?
Yeah, that's a fantastic question. So in the catalytic site prediction, we had, I guess it's basically 99% negatives versus positives. So we were pairing the data up and we tried to maintain not that imbalance, but a relatively imbalanced set. And that only actually became relevant in the LSTM. But for the contrastive learning model, it was more evenly balanced.
Okay, I saw another hand up. Where is the second question?
Thank you. You were saying you come from a computational background. How did you make that switch / the combination between the two?
Yeah, sure. So I got, so I did computer and electrical engineering in my undergrad. And during my undergrad, we had to do a research project, an honors research project, and I did mine on making software for looking at metabolic pathways. I didn't know what a metabolic pathway was or what DNA was at that point. I'd never really taken biology. And I guess I just got interested in it. And after working as a software engineer, my old research boss was like, hey, please come back. I've got a cool project that you should work on. And now I'm here and I work in the lab as well. So it gets you stuck. Don't ever do a biology project if you don't want to go down that track, because it's way too fun. And there's so many really cool problems, especially on the computational side, that haven't even begun to be solved. Like, you see all the cool protein design models, but that's just scratching the surface of the things that we can do. And I really think that, you know, moving forward, we're starting to get the computational power to be able to answer and uncover some of the most like unknown questions and unsolved questions that we care about as human beings.
Okay, thank you very much.
And we have one more last question here in the last row.
Okay, thank you. One last question. You talked about the use of agents in this whole thing, but you also said that the whole model wasn't that great at the time. So I wanted to ask, when you determined the threshold, and I said, okay, the outcome of the agent was good enough for your whole project?
Yeah, that's a fantastic question. So it was kind of a challenging problem for the agents. So what we were getting them to do was to take supplementary information and also the PDF of the paper, to go through that and pair specific information in figures with sequences that were in the SI, because that's typically how people in our field structure it, but obviously not all people structure their papers that way. And we were also judging correct as exactly sequence perfect. And so this is a very tough thing for even humans to do. Sometimes actually the LLM found mistakes that humans made, so I actually think it's quite nice to have both the person and the agent doing it at the same time. So we only got about 30% perfect extraction, but of the other 70%, it got most of the way there. So I do think that having a gold standard benchmark, so what we did was we went through manually, I think like 30 papers. There was about 15 of us doing it. And then we had that benchmark, and I think having a set that you really trust, and then you can say, okay, we're now going to make our agents go on another few hundred papers, which is what we did. We can then get an understanding of the mistakes that it's going to make, so that then we can go just look for those specific things as well. So I think it's also about understanding what the failure modes are, because these things aren't going to be perfect, but then not throwing them out because they're not perfect, but understanding the weaknesses and then working with them. So that's kind of how we approach it for our work with agents. Thanks.
Do we have one more question? Good. In that case, we are perfect in time. Thank you,
Well, thank you so
much
much.
for this talk. A big round of applause, please.
Yeah.